Hello, everyone, and welcome to today's broadcast, a keynote presentation, Molecular Mechanisms of Gene-X Environment Interactions, Possible Relevance for Prevention, Diagnosis, and Treatment of Psychiatric Disorders. It is presented by Elisabeth Binder, PhD, Director of the Max Planck Institute of Psychiatry. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Binder. I will now turn the presentation over to her.
Okay, thank you very much. I'm very happy to present some of our work in which we try to elucidate molecular mechanisms of gene-based stress interactions in order to get a better understanding of the pathophysiology of psychiatric disorders, maybe design novel treatment and prevention strategies. So the risk for developing psychiatric disorders is influenced by both genetic risk as well as environmental stressors. And these are especially impactful when happening early on, as early as during pregnancy in utero, but also during infancy and childhood. These stressors dramatically increase the risk for individuals to suffer of psychiatric disorders later in life. However, after experiencing such adverse life events, not everybody actually follows a risk trajectory, but there are individuals who actually remain resilient to these adverse life events. So in my presentation, I would like to present two main themes. One is that this long-term embedding of risk after being exposed to adverse life events can actually be mediated by epigenetic mechanisms. And the second is how genetic factors could actually moderate this embedding of risk. In my talk, I will actually focus on one system that I think is critical as an interface between the environment and the organism. And this is the stress hormone system. When we're faced with a threat, the system is actually activated and leads to the release of ACTH from the pituitary that then actually stimulates the adrenal gland to release glucocorticoids, the main glucocorticoid in humans being cortisol. Cortisol actually prepares the body for a fight and flight response and to actually be able to cope with the stressor. It is a hormone that reaches all tissues in the body. What is important about the system is that not only does it react to stress, but it's also the main system that actually shuts down the stress response after the threat is over. And this happens via negative feedback mechanisms at several brain levels that actually shut down the activation of this axis. So an important system at the interface between the environment and our body, and especially this balance between activation and inhibition is often disturbed in psychiatric disorders. A second reason for why I think this system is especially important to look at in the context of gene-environment interactions is that the main receptor for the stress hormone, the glucocorticoid receptor, is actually a nuclear receptor that translocates to the nucleus and binds to specific elements at the DNA to engender a transcriptome-wide response to stress. So via the glucocorticoid receptor, stress can actually have lasting effects on the cellular response, and this could be moderated by genetic variation. So the question is, how can genetic factors and environmental factors actually converge on specific genomic elements to convey mechanisms of gene and environment interactions? And for this, I'd like to point out the response elements that I just highlighted for where the glucocorticoid receptor is binding, termed GRE here in this picture. And glucocorticoid response elements can actually enhance or suppress transcriptional responses. With the activation of the glucocorticoid receptor and the binding to these response elements, not only is the transcriptional landscape changing, but these receptors can also engender a local epigenetic response. They do actually lead to changes that open the chromatin by inducing histomodifications, but by also reducing the DNA methylation content at these sites. So that with a prior activation of this receptor and binding to the response elements, one can actually alter the set point for the future transcriptional response. In addition to the environment being able to impact on the transcriptional response, but also on the epigenetic landscape of the target genes, genetic variation can actually also possibly moderate how glucocorticoid response elements actually moderate both the transcriptional response, but also the epigenetic response. So in this talk, I would like to give you some examples of how such interactions of genetic variation, environmental variation, can actually lead to changes in gene expression, also via changes in epigenetics. And I would first like to highlight an example of how stress can actually lead directly to changes in the epigenome. And for this, we actually looked at a cell model. We used a multipotent human hippocampal progenitor cell line derived from a human 11-week-old fetus. And this cell line can actually differentiate into adult neuron. It expresses synaptic markers. Prior publications have proven that glucocorticoids or stress hormone is actually effective in impacting these progenitor cell lines. And when we map the gene expression profile of these human progenitor cell lines, we actually see that they cluster with gene expression profiles from fetal brain tissue from 16 to 21 post-conceptional weeks. So they're actually a possible model for what's happening to the brain during pregnancy. And as I said before, pregnancy is a time point during development where there are a lot of environmental impacts can have lasting effects. So what we wanted to understand here is whether glucocorticoid receptor activation can actually induce changes in DNA methylation that in these hippocampal neurons while they're undergoing neurogenesis. And then to actually also understand whether such epigenetic alterations could indeed relate to the long-lasting effects induced by early life stress exposure in humans. So in our model, we treated these hippocampal progenitor cells at different stages during their development. So we treated them early on during the proliferation stage, but also while they were differentiating, and then harvested the cells. But what we also wanted to know is what happens if we treat them early during proliferation and differentiation, and then actually leave them alone, untreated for another 3 weeks to understand lasting effects. And this is depicted in the third pictogram. And we also wanted to understand whether the timing of the treatment with glucocorticoids matters. Is it early on during proliferation and differentiation, or after differentiation that actually matters for these epigenetic changes? And this is depicted in the last pictogram. And so we harvested the cells at these 4 different treatment time points, and then we measured genome-wide DNA methylation as well as RNA expression using Illumina array technology. So we first wanted to know, do our different treatments actually lead to differences in gene expression? So do glucocorticoids change the transcriptional landscape? And this is depicted here in this first slide here, where you can see that the most changes in gene expression actually happen when we treat during proliferation or proliferation and differentiation, and that the amount of transcriptional changes goes dramatically down after the washout. So we find over 5.5 transcripts that are significantly changed after correction for multiple testing across all these 4 time points. When we look at DNA methylation, we again see that treating during the proliferation stage actually leads to the largest amount of changes in methylation. Overall, we do see close to 30,000 DNA methylation sites to be changed significantly. And these changes go down in the other treatment time points. But what is interesting, that there seems to be a divergence of gene expression changes and methylation changes when we treat the cells early and then leave them alone for 20 days after these washout periods, where we have relatively low numbers of gene expression changes, but quite substantial changes in DNA methylation. And so we were very interested in really understanding the dynamics of the changes in gene expression as well as DNA methylation. So what we did first is we looked at the changes of DNA methylation across these different treatment time points. And we looked at all the ones that had significant changes above an even more strict cutoff than I just showed before. And we observed that the changes in DNA methylation actually clustered in 4 distinct trajectories. So we had, for example, the green trajectories where CpGs actually had reduced methylation over differentiation of the cells, but with the treatment, there was also always a bit higher methylation. And then there was an opposite pattern with increasing methylation with differentiation, where the dexamethasone or glucocorticoid treated cells actually had lower methylation levels across the whole trajectory here. And then there were trajectories that were not affected by the differentiation of the cells, but just affected by dexamethasone itself. What was interesting when we tried to find which pathways were enriched among these different trajectories, we found that the green and the red trajectory actually enriched for pathways relevant for cell proliferations and differentiation and developmental processes, whereas the more stable trajectories, especially the trajectory with the most pronounced glucocorticoid effects, actually were relevant also for transcription and regulation of transcription, which makes sense as the glucocorticoid receptor is actually a strong transcriptional regulator. Next to the pathway enrichment, I'm actually showing some examples of specific trajectories of DNA methylation of candidate methylation sites and genes, where you can see that a pattern of increased methylation is mostly followed by a pattern of decreased mRNA expression over time, so that we have a concerted change for some of these between DNA methylation and mRNA expression. What is interesting, but maybe a bit more difficult to see, is that at the last time point, when we treat after differentiation and have the washout period, there's actually relatively little changes in both DNA methylation and gene expression. And this is illustrated in more detail here for one candidate gene that we looked at. And you can see that on the upper panel, the DNA methylation actually is dramatically reduced with proliferation treatment, proliferation and differentiation treatment, and stays demethylated after the washout. But this demethylation is actually not happening when the cells are treated, when they're already differentiated. So there seems to be a developmental window that is important for when the treatment is happening to cause these epigenetic changes. And what is also important that we do see is that gene expression changes concur with early time points, but are absent with the quite dramatic changes in DNA methylation after the washout period, as I already showed before, across all the changes that we actually observed. So I really want to focus on this finding that we have quite profound and lasting changes in DNA methylation that are not accompanied by changes in gene expression at baseline. So we wanted to look at this in a bit more detail, and we actually took the locus of this FKBP5 gene and used targeted bisulfite sequencing to fine map the glucocorticoid response elements in this locus. And there are actually quite a few glucocorticoid response elements that are distributed in the proximal enhancers as well as the introns. And so I would like to give you an example of two intronic glucocorticoid response elements. And I'm showing each individual CpG within these glucocorticoid response elements and their methylation level after vehicle or dexamethasone treatment. And what you can see is that each CpG within that glucocorticoid response element actually shows this quite dramatic and lasting change in DNA methylation. So when we aggregate the finding across all glucocorticoid response elements, we do find that these elements actually are quite methylated initially. And you can also see in the lowest panel that the reduction in methylation actually seems to center right around the actually predicted motif for the binding for the glucocorticoid receptor, suggesting that it's actually active binding of the receptor that leads to these changes in DNA methylation. And it has been proposed that specific DNA repair mechanisms together with hydroxymethylation changes could contribute to this active demethylation. So I've shown you that glucocorticoid receptor treatment at specific developmental changes can lead to lasting effects on the epigenome with lasting decreases in DNA methylation, but that this is not followed by basal changes in gene expression. What we have shown before is that changes in methylation of these glucocorticoid response elements is not associated with baseline changes, but changes the set point for activations by glucocorticoids. So that in the demethylated version or unmethylated version, when introducing this element in a reporter gene assay, and this is depicted in the white box plot, you can actually see a stronger enhancement of transcription than when you insert a methylated version of this glucocorticoid response element in the vector. So that one could actually propose that this demethylation, while not leading to basal changes, would alter the set point for future activations with glucocorticoids. And what we wanted to explore is whether these early treatments or early exposures to the stress hormone could actually lead to poised states that would alter the set point of the organism to future stress exposures. And for this, we first wanted to know where we actually have these changes in DNA methylation. And for this, we looked at mapping the changes in DNA methylation to the Chrome HMM annotation of the roadmap epigenomic process, where the sites or regions in the genome are annotated for their function in being, for example, an active transcription start site or an enhancer that can be active or weak. So we actually annotated or looked whether any of our changes at the different time points would be enriched for specific enhancer elements. And what we observed is that the changes that happen during the proliferation phase are enriched for changes in active transcription, which would actually make sense since these are cells that are actively replicating. The second enrichment we saw were the changes that happened in both the proliferation and differentiation phase, but also remained after the washout, were enriched both in enhancers, but also in glucocorticoid response elements, sort of confirming that we get direct effects of the glucocorticoid receptor on the response elements, and that we're also changing enhancer function. But what was most interesting is that there was a subset of regulatory elements that were only enriched in those sites that changed only after, or only significantly changed after the washout period. And these were actually bivalent or poised transcription site or bivalent enhancers. Again, confirming that these lasting changes could be very important in not regulating baseline transcription, but the reactivity to subsequent exposures. And so this we wanted to test with an additional experiment. And so we repeated the experiment I just showed you before, but we added, after the washout, an acute stimulation with glucocorticoids with a lower dose. And so what we did see is that when the cells were treated with vehicle throughout and then had an active stimulation, we saw some changes in gene expression, and this is depicted in the box plot in yellow. When we treated the cells with dexamethasone and then did not stimulate, there was not a strong change in gene expression. But when they were actually treated early on, then had the washout and then re-stimulated, the extent of the transcriptional change which was larger than if they were just treated with an acute stimulation of dexamethasone but did not have this chronic treatment before. Again, suggesting that we poise the cells to react differently to a second exposure to the stress hormone. So these were all data that we gathered in a cell line, a human cell line, a human fetal cell line. And our question was, would the changes that we observe actually relate to changes that we observed in humans that are exposed to actual adverse life events and not just glucocorticoids? And so we first collaborated with McGill in Canada, who have generated data sets from human postmortem hippocampus of individuals who either died by suicide or control individuals, or individuals who died by suicide but also had documented exposure to child abuse during their early years. And what we did see is that we find a strong enrichment of the methylation sites that change in our cell model among the methylation sites that are also different between control individuals and individuals who either died by suicide, and even more pronounced if they died by suicide and had been exposed to child abuse, suggesting that there is at least some relevance of the findings of the hippocampal progenitor cell model to human postmortem hippocampus and of glucocorticoid exposure to child abuse. Since there are many more data sets who explore the same question of changes in DNA methylation after exposure to child abuse in peripheral tissue, such as peripheral blood, we also wanted to understand whether there's some overlap with data from peripheral blood here. And what we did see is that in 2 independent studies that observed DNA methylation changes in adults exposed to child abuse, we saw that there was actually also an enrichment of our sites in the peripheral blood data, suggesting that since glucocorticoids reach all tissues in the whole body, there might be some cross-tissue relevance of these effects, and these might be used for biomarkers in the future. So to summarize this part of the talk, stress actually changes gene expression, but stress also, after these changes in gene expression have subsided, can leave lasting epigenetic changes. So it changes the epigenetic set point. And with a second exposure to stress, these changes in set point could actually contribute to change trajectories to either risk or resilience to psychiatric disorders. So understanding how these set points are changing could actually help us understand to prevent psychiatric disorders. So I showed you this picture before of genetic variation, epigenetic changes, and gene expression changes, and how these are all orchestrating our response to stress. And I now want to show you how genetic variation can actually alter our gene expression response to stress and how that may contribute to risk for psychiatric disorders. So we wanted to explore such expression quantitative trait loci. And usually what you mean when you say expression quantitative trait loci are genetic variants that change the amount of basal RNA transcription. And we were not interested in basal RNA transcription, but what we were interested in was RNA transcription changes after stimulation with glucocorticoid. So basically, we're interested in scenarios where the genetic variant does not necessarily influence baseline transcription as depicted in the reddish box plot, but actually changes significantly the extent to which gene expression is changing after stimulation with glucocorticoids here. And to assess this, we actually did an experiment where we used 160 male individuals. We took their blood at baseline, extracted both DNA and RNA, and we then gave them oral glucocorticoids and then measured their gene expression in peripheral blood again 3 hours later. And with the DNA, we measured genome-wide SNP variation, and with the RNA, we measured gene expression both at baseline as well as after the stimulation. And what we wanted to understand is, are there genetic variants that actually moderate the local change in gene expression after glucocorticoids? And when we ran this analysis, we actually identified close to 4,000 eQTLs, meaning 3,600 SNPs that moderate the extent of change of close to 300 transcripts after glucocorticoids. And most of them fell in this pattern that I showed before, with very little changes at baseline, but prominent genetic impact on the stimulated gene expression. So we wanted to understand what are these SNPs that have little impact on baseline transcription, but actually change the transcription after basically a mediator of the environment. And what we found is that these genetic variants that alter the transcriptional response to a stimulation, to a stress stimulation, are actually much more distant from the transcript than genetic variants that influence baseline gene transcription. We also found that this long-range regulation could be mediated by changes in the 3D structure. So we found direct chromatin interactions of these SNP sites with the transcription start site, both by genome-wide and targeted approaches. We saw that the SNP sites were actually enriched for binding sites using chromatin immune histochemistry precipitation with the glucocorticoid receptor, but also for predicted transcription factor cofactors for the glucocorticoid receptor. And what we also found is that these SNP regions are actually enriched in enhancers, not only of peripheral blood, but also of the brain. This would suggest that there are SNPs that alter the genomic response to stress. They seem to be distinct from SNPs that alter baseline transcription. And so the next question was, do these SNPs actually increase the risk for stress-related disorders, such as depression, for example? So to get at that question, we used genome-wide association, SNP association data for major depression. And back when we ran this study, there were no genome-wide significant hits for major depression so far. So we had to assume that there were some true genetic hits in the nominally significant associations. And our question was, would our functional SNPs be more often than chance be represented among the nominally significant SNPs associated with major depression? And this is what we wanted to find out here. So we first looked at the expected number of either random SNPs or SNPs that influence baseline transcription with SNPs that are associated at nominal significant level with major depression. And these are the distribution in blue and in light gray. And then we tested that, compared that to the actual number of SNPs that overlap, which is actually this red arrow, and found that never did a random distribution reach the actual number of overlapping SNPs, suggesting that there is enrichment of our glucocorticoid response e-SNPs among SNPs associated with major depression. So we next wanted to confirm this association with depression in an independent sample. And for this, we took the overlap between our functional SNPs and the SNPs associated with depression in a large GWAS, and actually looked at SNPs that would uniquely identify this overlap and summed the number of risk alleles in this overlap. And we calculated that some of these risk alleles in an independent sample of depression cases and controls. And what we found is that there was a significant shift in the distribution of these risk allele sum scores in cases with depression, so that they had slightly higher numbers of these risk alleles than controls, confirming again some association of these variants with major depression. So since these SNPs actually react to glucocorticoids, we wanted to know whether these associations would actually be unmasked, especially under conditions of early adversity. And so with this, we looked at another cohort where we actually had information of adversity during childhood. So we had individuals with high adversity, depicted the red line, medium adversity, the green line, and low adversity in childhood being the blue line. And then we looked at their genetic stress profile score and whether it predicted mood and anxiety disorder. And what we saw is that the genetic profile score actually only predicted risk for mood and anxiety disorder when these individuals were exposed to high levels of early adversity, suggesting that the risk-conveying properties of these SNPs are only unmasked when individuals are exposed to stress early in life in the term of a gene-environment interactions. So these SNPs actually not only seem to predict mood and anxiety disorders, but also are associated with endophenotypes associated with the stress response in the brain. For example, like amygdala reactivity to threat or cortisol reactivity to mental stress. So I have shown you that there are genetic variants that alter the first transcriptome response to stress within a few hours. These SNPs seem to moderate neurocircuit response to threat and also risk for mood and anxiety disorders. So the question is that trauma and stress is not only a risk factor for depression, but also for a range of other psychiatric disorders. So we wanted to know whether maybe our SNPs are also enriched in genome-wide association studies for other psychiatric disorders. And we saw a strong enrichment also in SNPs associated with schizophrenia, for which is also exacerbated by exposure to early adversity. And what is interesting is there seem to be a number of functional genetic variants that are responsive to stress that are associated not only to depression, but also schizophrenia and even bipolar disorder, suggesting that there is a cross-disorder type of risk that could be conveyed by these stress sensitivity genetic variants. So an enhanced stress vulnerability without disease specificity. So I talked a lot about the genetic variants, but what about the transcripts that they're influencing? What was interesting is that when we take all the transcripts that are influenced by genetic variants that alter their responsivity to stress and are associated with either depression or schizophrenia, we actually found a highly co-expressed network of transcripts that actually shows enrichment for specific pathways that have been related to psychiatric disorders as well as stress, such as endocytosis, that's important for neuronal signaling, as well as cytoplasmic ribosomal proteins. So a network of transcripts that are co-expressed and co-regulated by stress, a glucocorticoid responsive co-expression network. And so we wanted to understand whether this co-expression network of transcripts would actually be affected by exposures to different social experience, both positive as well as negative. And for this, we actually used an animal model that combines positive early experiences, such as early handling, or negative experience, such as limiting nesting material. And then these animals with either positive or negative early experiences are then randomized again in either having a positive adult social experience being housed together with an overrectomized female, or experiencing chronic social defeat stress over a certain amount of time. So we have conditions of animals receiving positive environment throughout their lives or negative environment throughout their life, or having mixed environments. And then we looked at different brain regions that are important for stress regulation and actually tested the gene expression of these over 60 transcripts that actually have 55 mouse orthologs using targeted RNA sequencing. And so what we did observe is that there were some, but not very strong, main effects on gene expression. But what was more pronounced is that the co-expression network pattern actually changed with the different types of social exposure, the combination of positive versus negative. What we noted is that in the paraventricular nucleus, we actually saw that there was a strong network structure of these genes in a positive social environment, an enriched early environment, and this network structure disappeared with exposure to stress. And the network structure was not present with positive environment in the ventral CA1, a part of the hippocampus, but actually seemed to emerge in stressful conditions. So in the absence of strong main effects, there seem to be social stress dependent changes in network properties in a brain region specific way. So this is another way to look at that, how the network properties actually change with the brain region as well as the different stress conditions. And so the first thing we noted is that the network structure was actually closest for the hypothalamus, the paraventricular nucleus, for the enriched early environment, so being very close together. The second thing we noted that there were hub genes or central genes that were common for both the low stress condition in the hypothalamus as well as the high stress condition in the hippocampus, suggesting that there are concerted changes of these networks across different brain regions and different conditions. So there seem to be shared central network genes of the two brain regions and the two environments. And this slide here depicts the network in the hypothalamus with early enrichment, where we have these strong networks, and highlighted are specific central genes that appear both in the paraventricular nucleus for the early enrichment, as well as the ventral hippocampus for the stressful stressful environments. So this suggests that there are SNPs that alter the first transcriptome response to stress, that moderate neural circuit response to threat, which leads to an increased risk for mood and anxiety disorder. And the question is now that we think that these SNPs actually alter the co-regulation of stress response networks and thereby circuit function that predisposes to risk for psychiatric disorders. So just very quickly, I showed you that stress can change or can be moderated, have its effect moderated on gene expression. And this was done in peripheral blood with some relevance for the brain. And that stress can also directly impact DNA methylation properties of neurons. And when we actually combine these two data sets, what is quite interesting that we get a strong overlap of the sites that show these changes in methylation and the SNP sites that actually moderate the glucocorticoid induced response. So there's an overlap of genetic as well as epigenetic risks that actually come together and that could further contribute to these gene-environment interactions. So in the end, I showed you some very specific examples of how genes and environment can actually interact. I picked specific examples for the genome, looking at single nucleotide polymorphisms, and of the epigenome only focusing on DNA methylation, but it needs to be acknowledged that there are many layers on the epigenetic side as well as the genetic side, and also on the proteome side that were not mentioned. I also wanted to convey that stress early in development can actually lead to alterations of lifelong risk or resilience trajectories, possibly by leading to changes in epigenetic in the epigenome of regulators or enhancers of stress responsive genes, thereby changing the set point for future stress responses. The question is now, why would we care? Can this actually lead us to important insights for the clinic? And we actually have identified a candidate that is regulated in that manner, that is called FKBP5, where early exposures leads to an epigenetic de-repression that is associated with future risk for psychiatric disorders. And this molecule can be targeted by small molecule antagonists, and in animal models, this seems to have a stress reducing effect in animals, and so could be a possible strategy to improve resilience in individuals exposed to risk. And with this, I would like to end my presentation and thank a large number of coworkers and collaborators worldwide. Thank you very much.
Thank you, Dr. Binder, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, what is the presumed mechanism of how glucocorticoid receptor activation leads to epigenetic changes?
Yeah, this is a very important question. I think there are two different sets of mechanisms that can contribute to that. One is actually the glucocorticoid receptor binding to the response elements in the DNA and leading to local active demethylation. And here some papers have proposed the involvement of DNA repair mechanisms and maybe also of introducing intermediate steps such as hydroxymethylation via the 10-11 enzyme family. But this has not been proven yet and is under current exploration. So a receptor-mediated active demethylation. The other effects that we do see seem to be indirect effects that may be mediated by genes that are activated or repressed by glucocorticoids that then in turn could lead to changes in the epigenome.
Thank you. Next one is, do polygenic risk scores for psychiatric disorders interact with early adversity to predict risk?
Yeah, again, a very important question also with the now ever increasing number of genome-wide association studies that we do have for psychiatric disorders, and the knowledge that both genes and environment contribute. There actually have been quite a number of studies that have explored how polygenic risk from these case-control GWAS would actually interact with environmental risk factors. And there are different findings. For example, the polygenic risk score for major depression does seem to have additive effects, but not to interact with early adversity. Whereas, for example, the polygenic risk score for schizophrenia seems to moderate the impact of obstetric complications on the risk to develop schizophrenia further on. So what is important is that we need to look at these genome-wide association study polygenic risk scores, but also to maybe explore other functional polygenic scores, like I showed in my presentation, where we have a more funneled hypothesis about SNPs that moderate the stress hormone receptor impact, whether these would actually then interact with early adversity.
Okay, thank you. Looks like we have time for one more question. Could we develop biomarkers for prevention using these approaches?
Yeah, so this is again an important question, and often epigenetic profiles are distinct between different tissues. That means the brain and the blood. What we do observe is that glucocorticoid activation actually leads to some extent to overlapping changes in blood and brain. And so that maybe changes of the DNA methylation pattern in peripheral blood could serve as a sentinel for what is happening in other tissues, including the brain. And so I think understanding how different impacts, both genetic as well as environmental, impact on the epigenome in a cross-tissue manner can actually also help us to develop better biomarkers for stress-related risk.
Thank you. I would like to once again thank Dr. Binder for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 27th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.